Okay.
But I guess
Let's have a look. We can always change if you've been through them. It's just going to be a waste of your time, but let's take a look.
2015. Okay.
Recognize this?
We've probably done all of the papers.
Like some of the older ones. I think I probably have done this, which is 2015.
Okay.
I've done all of 2015 papers.
Okay, shall we let's pull up a different paper then.
Okay.
But
you have to go back to like
ancient
2012-ish.
Okay, I mean we can do that. We'll just there will be questions that we'll just skip because they're not going to be relevant.
If you okay.
Yeah.
Should we do? Are we going to do one paper, or do you know?
We usually get through two.
Cool. Should we do then one old paper and then one newer paper?
You can do like more of the new paper if you want to. Then.
Okay, cool. Well, you know, you can do it. You know everything anyway. It's okay. He's my brother, so let's say that.
Let's get another paper up then. What's the easiest way? This computer to find
Paper.
Yeah.
Right.
And then we can do the 2016 one because this is quite relevant.
Cool.
I'm just gonna pick over like one from 2012.
Just take a quick look at it because we
we might. Yeah.
Looks familiar. Yeah,
we've
we've
done
done
this.
that.
You know what is interesting though? When you go back really far back, some of the questions are so similar to the 2017 paper. Like I realized that today looking at some P5s, and I went to 2009.
Wait, I think we've done this too.
And it was like such a similar question.
No. Actually, I don't recognize this.
that.
Yeah,
Okay,
let's do this.
Let's do this.
Get the marking scheme as well, so that
Okay.
we can mark you guys as you can.
Should I get the mark scheme for the other paper?
I've got the marking scheme for
Okay.
2016 here printed. Yeah. Is everything good?
Yeah. Yeah, we're good.
Okay. Cool. So,
Wait, this mark scheme.
oh. Sorry. Let's not just scroll down. Yeah, this is it. Okay. So, we are looking at bacterium. It's the easiest way to zoom out a little bit. Okay, so just quickly tell me how you do this. We don't have to do it. Are you both comfortable? I know Mark's comfortable with this kind of question, Grace. Are you comfortable with it?
So, I measure this, this length. And then once I have that, I know that is equal to 0.5, what is this, micrometers. And then I convert my centimeters into micrometers, which is, it's centimeters and then millimeters, 10. Do I go down 10 millimeters and 1 centimeter?
Yeah, 10 millimeters.
No.
It is 10 millimeters.
Okay.
I would just avoid centimeters. Like
Okay.
you can measure with your ruler in millimeters, and going down from meters to millimeters to micrometers to nanometers, it's always steps of 1,000.
Okay. Okay, cool. So I convert it into micrometers, and then I know how many more I can divide. So if I have like 100, I divide that by this number, and then I get the magnification.
Yeah, good.
Okay.
You know, so when you're doing this question, there's 2 marks. One is for getting the answer right. The other is basically for writing an equation.
Which
you just described to me, and it's correct, but easiest way to get these marks is just to write the equation. It's just letter form. You don't.
Is it the actual image over the other image? I don't know exactly.
So
What do we call the other image?
M is equal to I divided by A. It's, yeah.
Magnification is equal to?
I, which represents the image, and then the A is
the actual size.
Mhm.
Okay.
So for this one, so if I told you that scale bar is 20 millimeters, could you do the calculation?
So that's how many micrometers I have, right? And then I need to divide that answer by 0.5.
Good.
And then
What would that be?
4,000.
Yeah, good.
4,000. Okay, and then I get a magnification.
Good. Yeah, and you just put it in that box. Now that's for calculating magnification. They can ask these questions in 2 other ways. Either one other way. So they could show you a scale bar and give you a magnification, and ask you to calculate the size of an organelle or something like that, and you use the same formula, you just rearrange. Exactly. Okay, cool. That's fine. What's the best way to do this? I reckon we'll just look at the question, and then afterwards we can look at the markings. Okay, Mark, you do this one. Oops.
Okay, so the endoplasmic reticulum, the Golgi apparatus and nucleus.
Okay, good. So what we're doing here is comparing bacteria to animal cells and just listing organelles that are in animal cells and not bacteria. And that's right. Any other organelles you can think of present in animal cells, non-bacterial?
Vesicles?
Yeah, you can probably get away with that. You can get some kinds of vesicles and bacteria, but let's see what they say. Okay, so yeah, they focus on
Like
the
nucleus
DNA.
is definitely like,
Yeah.
The obvious ones.
and then everything with double membranes is also.
Mhm.
Okay. Cool.
Is that the type of bacteria we have?
It is. Or, sorry, no, it's not. What did we have earlier?
These are the same, right?
Yeah.
Yeah.
Okay. Maybe they're not fully developed yet, or or is it because we're looking under electron microscope and they've, because they're dead, they've, I don't know, deformed?
You're on the right track. So we're thinking about what we're literally looking at. We're not looking at bacteria in a 3D environment. So if I told you that they are all rod-shaped, these are all rod-shaped bacteria, why might these two look like this in the EM that we've taken?
They've lost some of their insides.
Not quite. Well,
It shows like a plane of the specimen. So that might be a rod, but you can't see it in the
yeah, good. So what we have is something like this. I can attempt to draw this in 3D.
This is a weird question though.
And so you had 3 bacteria, and they were arranged like this in a 3-dimensional space. And then the section
Oh, okay, I get it.
kind of went like this. And so we cut these in their stem laterally, but this one we cut longitudinally.
Oh, okay, I get it.
Yeah.
I get it, yeah.
Cool. They don't ask that kind of question.
But they might, because like they might. Sorry, now that I've seen that other paper, I feel like they're gonna take old questions.
Yeah.
Not just, that's just.
Okay. Let's just check the markings here for you. So, good, yeah. Depends on angle cut, /cut in different planes, /end view. I mean, you could make up some biological explanations as well. The bacteria could have become desiccated in preparation. That's feasible, but it's not the most obvious reason. Yeah. Okay.
It's me, right? Okay. Glycosidic bond.
Okay.
So the plant cells, they'll break away from each other. So will the vegetables fall apart?
Okay. So you're thinking of the actual vegetable part of the plant, so the actual carrot or the potato. But if you think about the plant, sort
It'll
of
be like the stem.
yeah, think about it in sort of AS/A biology terms. We don't really focus on the sink, which is the carrot or the potatoes, but think about the structure of a plant and how, if you start breaking those cells apart, how the function of the plant could be limited.
So could this mean that you have like the xylem and the phloem would be affected and you won't have as many nutrients, like, or would you have more because the xylem, I don't know.
You're on the right lines. So thinking xylem, phloem, transport vessels, they're made up of cells. If you start breaking those cells apart, what functions do xylem and phloem contribute to?
The xylem contributes to living transport.
Good.
And the phloem to things like sugars and the nutrients that the plants need, I guess.
Good. And so if we compromise cell adhesion, then how might that affect transport of water and assimilase?
Oh, okay. So less water is transported to cells that need water to become turgid, so wilting.
Yeah, good. So you get wilting.
You have less photosynthesis occurring. No, sorry, that doesn't. You have the assimilates are not being transported to the sinks, so less respiration for those cells, or growth or storage.
And in terms of photosynthesis, So what mechanism does transpiration depend on?
Evaporation.
Yeah, good. And that requires the movement of water, right? And if we're compromising the xylem, then we're compromising the plant's ability to transpire. So the photosynthesis itself will also be limited.
Okay.
Let's have a look at the marking scheme. Okay. Except need two likely vaccines. Okay, cool. So this is actually just talking about the vegetables. So you would have
I think this is very weird.
you would have gotten the marks for talking about vegetables becoming soft, mushy, and edible. Turgidity is a good point there. Yeah, okay. I would say don't worry about this because if you look at it's always the last 5 years that are the most relevant. And, between now and tomorrow, these are definitely far from the kind of questions that we see year on year for the last 5 years.
I'll remember that. Soft vegetables.
Okay, sorry. Okay, cool. This is quite relevant. So they could ask you this. Talk me through this, Grace.
Okay. I haven't done this in a long time, so I might be wrong.
Okay.
So I know you have. Okay, so I know you have the sand, which is the first one in the upper left, I think, and does that create an electrical impulse, which causes then the atriums to contract, which then passes through the AVN, which is sort of in the middle, and that causes the ventricles to contract, and then the electric that charge goes back up to the.
Okay, you're sort of on the right tracks. If I drew the heart here, Mark, where's this synovial node?
On the right, in the wall to the right atrium.
Yeah, so somewhere here. Do you guys get how I've drawn this heart?
Yeah.
It's as if I'm looking at somebody and I've opened their chest and there's their heart. So the right atrium would be up here, even though it's on our left. AVN is found in the right atrium. It conducts action potentials across the myocardium of the atria and towards the atrioventricular node, which is found at the, as the name suggests, border between the atria and the ventricles. Okay, good. So what does the atrioventricular node do?
It delays the electrical excitation wave by 0.1 seconds. So it prevents the atria and the ventricles from contracting at the same time.
Good, and that's really important. And what else does it do in terms of the direction of the conduction?
It passes it on to the Purkinje fibers.
Good.
Conduct.
Yeah, exactly. So between the ventricles is this fibrous septum which divides them, and in that fibrous septums are these Purkinje fibers that go down the septum and then go up from the apex of the heart. And because those fibers are in the fibrous septum, they don't start the contraction until they come out at the bottom of the heart. And that's why the heart beats from the bottom up. So when it beats, it squeezes at the bottom as it's going.
So does the AVN delay the charge and pass it to the?
Purkinje fibers and the.
But that charge, it doesn't actually, nothing contracts until it reaches the bottom.
Yeah.
Okay.
That's detail that I wouldn't really worry about, but it's helpful for our understanding. Because you have that contraction of the heart at the bottom, it pushes the blood up towards the pulmonary artery. And
So it contracts in this. Yeah.
Same direction. Yeah. Good. Left AV valve, Mark.
So okay. It opens during atrial systole when the blood pressure of the left atria is greater than that of the right atria. The left ventricle, and it closes to make sure that blood doesn't flow in the wrong direction during ventricular systole when the blood pressure of the left ventricle is higher than that of the.
Wait a second. So it's open when the blood's going from the left ventricle into the left atrium, right?
When it goes from the left atrium to the left ventricle.
What's the valve between the left atrium and the ventricle?
The left ventricle.
Oh, so no, wait. So the blood goes from the atrium
Yeah.
to the ventricle, which goes out into the aorta. And
Yeah.
this one's the one between. Okay.
Yeah.
Cool. Happy with the concept of valve shutting when the ventricles contract, etc., etc. Okay, cool. In answering these questions, I mean, they really want to give you the mark. So, although it's saying describe the function, always briefly say about the structure or location. You know, you'll pick up a mark. We'll look at the marking scheme now, but I predict that they're going to say there's going to be a mark for saying that the atrioventricular valve is between the atrium and the ventricle, and it, etc., etc. Sinoatrial node is in right wall, that kind of thing. Let's see. I know they were much more stringent back in 2000 and whenever this was. So yeah, they're only giving the marks here for function. But yeah, we've covered these points. So acts as a pacemaker for the sinoatrial node, releases a wave of depolarization. The fact that it causes atrial systole. And then when we look at the AV node, as you said, it delays the impulse, it sends the impulse to the Purkinje fibers and bundle of His. And it allows atrial systole to complete the full ventricular system. And finally, for the valve, stops backflow of blood from ventricle to atrium, allows one-way flow of blood. Yeah. How familiar are you with this kind of graph? Okay.
I've seen it before.
All right, so they've pointed to various parts of this, and they're just asking you to match the points to the correct statement. So if we start with the first one, then left AV valve starting to open.
Is it A?
Why do you say A?
Because the blood is flowing from the blood is high, the blood pressure is high in the atrium and low in the ventricle, so it wouldn't be A. It's going from high to low pressure.
Pay close attention to this, because it'll help you work out.
Okay. So is it G?
Why G?
Because the pressure in the ventricle isn't high as yet, and the pressure in the atrium is not high either, but...
So the question is when the left AV valve starts to open. So if you imagine the valve sitting there, here's the atrium, here's the ventricle. When it starts to open, what's going to happen to the pressure in the atrium?
It's going to decrease.
Good. So at what point are they pointing at where you can sort of see that happening?
And then the blood pressure of the ventricle will start to increase, will it, as this starts to open?
So it's a bit complex because while this is happening, it actually causes filling of the ventricle as well, but the ventricle is in diastole, so you don't get a huge amount of pressure increase in the ventricle. The important thing to know here is that when that AV valve is opening, the pressure in the atria is going to fall, but it still has to be higher than the pressure in the ventricle to cause the blood to flow. So at which point is pressure in the atrium falling, but also it's still higher than the pressure in the ventricle?
At C?
Yeah, so C is the answer. Does that make sense?
What's happening then here?
Okay, and Mark? Do you want to explain what's happening at F and G?
Okay. Well, I know that at G, so the ventricle is starting to contract. Ventricular systole is occurring, so the blood pressure increases, and then it flows to the aorta.
Can I ask, is this the blood coming into the atrium here?
So when the ventricles contract and blood is going out of the ventricles into the aorta, the AV valve is closed, so that blood cannot go into the atria, but you might see a slight pressure increase in the atrium because of the force of the contraction of the ventricles.
Okay.
So the way these graphs are made is they put pressure catheters into each chamber and they just measure the pressure of the blood. So some of those changes in pressures, I wouldn't worry too much about the small fluctuations like this and this. Instead, look at key points where pressure is spiking or, you know, atria is higher than ventricles, that kind of thing. It doesn't occur for a very long period of time and it's quite stable, these changes. They're not going to be affected by the way you're measuring it. Whilst these kind of fluctuations aren't. It's going to be there.
Okay.
Okay, let's go down to the next one. So, starting to close. Yeah, go on.
No, is it okay? Wait, oh, okay. G.
Yeah, why does it have to be G?
Because that's the point where the pressure of the left ventricle exceeds the pressure of the left atrium.
Yeah, good. So for it to actually close, pressure here has to be higher than pressure here.
Okay, I get that.
Left ventricle starts to contract.
And that G as well?
Yeah, good. Blood remaining in left ventricle.
B.
Why?
Because that's the moment that ventricular systole stops.
Good, yeah, so all of the blood is emptied to the aorta at that point. And out of curiosity then, where is the blood pressure that we measure? The blood pressure cuff on that graph.
Doesn't it have to be like the highest? Because it's the easiest to measure.
Would it be too high if it's the highest? Would it? I think it'd be.
Wait,
before it
is
goes.
it even that we don't measure? Oh no, actually, okay, go.
Actually, so when we like put a,
it's the.
It would.
It's the ventricular system, like it's the.
After this, so at B, because aren't we measuring the blood in the pressure here?
Yeah.
So it would have to be after.
Oh, okay, that makes sense.
So you've got the ventricle contracts and then the aorta pulses, and that pulse wave will carry itself through all the arteries in the body, including the one here.
And that's
what we measured, yeah. And the waveform will be exactly like the waveform in the aorta. So it looks like that.
Would the pressure be the same?
So it's not as accurate and sometimes when you need a very accurate measure of blood pressure, you will put a catheter in the aorta and measure it there. But it's quite accurate. Yeah, it's not the fact that it's true to real blood pressure that's important. When you're measuring blood pressure, the most important thing is knowing what normal is and whether you're going above it or below it.
Okay.
Having the exact absolute measure.
But what if you don't know if what that person's normal is?
Yeah, that tends to be the case. So it becomes, you have to interpret it in the context of what we assume, let's say, the blood pressure for a 20-year-old person with that BMI should be. And that's how we assess it. Unless the person's in hospital, in which case you can just measure it and then you get an idea of what their normal is. Okay. Next question.
Okay. That's the epidermis, right?
Yeah, good. Just talk me through the different layers then.
So that's the epidermis. That's the Palisade mesophyll cells.
Mm-hmm.
These are air spaces, and those are also mesophyll cells, right? But they aren't Palisade.
Yeah, what do we call them?
I can't remember.
Spongy.
Spongy.
Yeah, because of the air cells.
Cool. And is that a stomata?
Yeah, good. That's stomata.
And that's the lower epidermis at the bottom. Good.
So use label lines, letters X, Y, and Z for formal features. Okay, you said cellular epidermis. We talked about Palisade and Mesophyll. Guard cell?
It's the one, the curvy one on the stomata.
Yeah, good. You want to do this one?
Okay. So water moves down the constant water potential gradient into the cell wall. Then evaporation of water occurs, which creates saturated airspace. And then the water diffuses down the water vapor diffuses down the water potential gradient to the open stomata.
Okay. You're right, the points you made are correct, but what they're asking you to do, if you go back to the question, is describe and explain it, how water moves from inside the leaf to point Q, or at point Q to the atmosphere during transpiration. Yeah, actually that's fine. Yeah, that's fine. When you were going through your explanation, your points were correct, but I would have maybe explained it in terms of during transpiration, stomata open, and then talk about it from that perspective. But if we take a look at the marking scheme, I'm sure that they'll give you all the marks for the points you made. Numbers of it, so yeah. Good.
Can I ask something?
Mm-hmm.
So you go from when it's going up the xylem, the water, it then gets it then osmoses into, does it, so when it gets here, how does it osmoses down its water potential gradient from the xylem into, I guess, here, and then it osmoses into the air spaces, and then it osmoses out of the cell onto the surface area, and then it evaporates.
No, so the evaporation occurs when it's exiting the mesophyll cell.
Okay.
So you have liquid water in the plant, in the leaf itself. So it's at this point when the water molecule crosses the cell wall that you'll start to get liquid H2O formed. And
Don't we always have liquid?
Yes, you do always have liquid, but I think that's a good question. When it's moving through the symplastic and apoplastic pathways, I think the distinction between liquid and it would still be liquid actually. But the point is evaporation occurs as it exits the cell. That's something you need to know.
But wouldn't it be
Water vapor.
gas then?
It's water vapor when it comes out. So like lots of water vapors.
So here it's a gas.
Yeah, so
Yeah,
like
it's
all
gas.
here, like it flows from
liquid.
this island and like as like water, and then it
Evaporates into the
yeah,
airspace.
Yeah, and then
Oh,
And then-
sorry. Yeah.
Okay,
I've
and
confused
no,
it.
it's okay. And then it diffuses out of the cell,
Yeah.
and it's gas, completely gas.
Yeah, vapor, yeah.
Okay, cool.
Okay, talk me through this.
So if it's in a sunny position, it's trying to, I guess, reduce transpiration because it doesn't want to. Oh, maybe the fact that there aren't many stem guard cells means that it's trying to, even though it still needs to photosynthesize, if it has a stomata open, then it's going to lose a lot of water quickly. So it doesn't have that many stomata.
Okay. Yeah. That could be one of the mechanisms. Although to do that cellularly is quite complex, but that's good. What other features of this plant suggest that it's chromosomal?
Is it because it doesn't have very many air spaces that they're trying to reduce water loss?
Okay.
Um,
What do you think, Bob?
there's a large layer of Palisade mesophyll cells, and the stomata is closed, and I don't know if I would say this, but there's only like one stomata, so out of the picture we can't see that there's a large number of stomata present.
Okay. How about the epidermis?
There's a waxy cuticle that's thick, so thick waxy cuticle.
Yeah, good. We can see cuticle over the epidermis. And what's the function of that?
So sorry, if we didn't have that layer, we'd know that there's no waxy cuticle.
Yeah, that's
Okay.
That's like
is
wax.
the waxy cuticle.
Okay, cool. And that makes it thicker or harder for water to get out.
So it prevents. Well, it doesn't prevent, but it limits that osmosis of water out of the surface of the plant.
Okay. And can you say that the stomata is closed? Like, does that
Yeah,
count as okay, cool.
Yeah,
Because you could open the stomata
that is true.
and then
It's not the best worded question, to be fair. What are you saying, Mark?
No.
You were-
That's good.
Okay, let's see what they say.
Yeah, no stomata at the upper surface. That's a good one.
Yeah. In the curriculum now, though, I don't think it talks about stomata ever being on the upper surface, does it?
I don't think so.
But yeah, that's a point that could be made.
But it like makes sense.
why would there be no on the upper surface?
Why
Well,
would
here
there-
it makes
there
sense
isn't.
because
There is not.
here it makes sense because obviously if
It's
the
a
sun is
zero.
shining, but why in general? Is there in general not many stomata on the upper surfaces?
Yeah, in general, there aren't many stomata on the upper
Because
surface.
of
Because
the
of
sun?
sunshine, exactly. Yeah. And the effect that would have on evaporation.
Mhm.
Okay, talk me through the scripts.
Okay. So at the start, I can say maybe till 1996, there's an overall increase, or maybe actually 1997, that there's an overall increase of people that are affected with HIV.
Okay.
And then I can say it starts to decrease from there on. I could probably mention some data points, like for example, it's where it does turn from an increase to a decrease.
Okay, cool, that's right. When it comes to these questions, this is quite a nice one because the data isn't doing much, but you could be given one where, there's multiple different data points that are going all over the place and they want you to describe it, they'll give you 5 marks. And it can feel like you're getting, sort of dropped in the ocean because there's so much to talk about. And the way I would always structure it is, and this is how they'll list it in their marking scheme as well, take a step back and just state the most obvious things you can see. And these things you can potentially miss if you just go into describing exactly what the data is doing. So the first thing I would say for this is the number of people who have HIV in total has increased from 1990 to 2008.
Okay.
And that's such a key point for whatever this data is going to be. And potentially one of the next questions it's going to say, why has it increased between 1990 and 2008? So always take a step back, mention the most obvious thing, and then go in and just describe the data as you go along. And that's exactly what you did. So between 1990 and 1993, there was an increase in the number of people at a steady rate. That increase continued, but the rate at which it was increasing started to decline until a peak in 1997. And then from 1997 to 2008, there has been a slow decline in the number of people with HIV.
Okay.
But yeah, that's good. Suggest possible explanations for the decrease in the number of people newly infected with HIV. Okay, Mark.
Okay. So this could be due to more contact tracing. It could be due to.
So what do you mean by contact tracing?
Well, so somebody has AIDS, then they can contact all of the people that that person has been in contact with. Yeah. And then education about the transmission pathway. So they know how to use condoms and stuff like that. So more about knowledge about the transmission pathway for drug users not to share unsterile needles, and better drugs to prevent the mother to transmit the virus towards the child.
Good.
Testing for blood for HIV antibodies.
Good. In general, the most effective thing in decreasing the number of people with HIV in developed countries is increasing diagnosis, because you might know a huge problem with HIV is that it causes a very minor illness at first, and then it can have a latent period for an average age of up to 15 years before the final decline into AIDS. And the reason we had, even after we figured out HIV was the causative problem, the reason we still had a huge increase in the number year on year was because we still couldn't diagnose people who were infected. On average, about 25% of people in the UK with HIV don't know they have HIV still today.
That's a lot of people.
And that's sort of an indication to say how much of a problem diagnosis is. So definitely talk about that. All the other points you made were good. You can talk about developed countries versus developing countries, that kind of thing. Lots to talk about on that.
When you, so for example, if somebody gets HIV from a blood transfusion, how likely are the chances that you will be infected by HIV if you're having, is it 100%? So
Pretty
like,
much 100%.
oh, so if somebody, or if you're having sexual intercourse with somebody else, it's almost 100%.
Pretty much 100%, yeah. So if the virus gets into the bloodstream.
It's all about like fluid exchange.
Yeah.
Right. So if like the infected person's fluid, then you have the virus.
So the virus is everywhere, pretty much in an infected person.
In an infected person, it's in the blood, and so to infect somebody, you need to get it from your blood into their blood, and it will be pumped out into excretions, mucus excretions, and then if those mucus excretions may go on to vascularized tissue on the other person, they can diffuse, and if the HIV gets into the capillaries, makes its way back into the bloodstream, it can start colonizing lymphocytes. It's not, so sex and IV drug use is like a huge problem, very easy to transmit. Other fluid interfaces, I mean, it's not going to transmit stuff like, kissing.
Why not? If it's in
The blood.
yeah, but if it's in, would it not be in somebody's saliva?
It would be, but it's all about viral dose. So the amount in the saliva is so much less than the amount in the blood. And if you kiss someone, the amount of their saliva that's causing molecular exchange on your mucosal surface is also very, very low compared to blood transfusion where you're like immediately putting it in, or sex, which also has high rate of transfer over those mucosal surfaces. So that's, I think on average, it's something like, if you wanted to give somebody HIV through saliva, they'd have to swallow a gallon of it, something like that. So it's not going to happen.
Pretty mad.
Yeah. But, okay, any other questions with that? Okay.
Is it, I guess it's like standard deviation, but I don't know why it needs to be included on the graph. I guess you can't have, I know you can't have an exact estimate because you don't know, obviously, how many people actually are infected.
Good, that's the answer to the question. Okay, sorry. Do you want to do this one?
Oh, okay. So I think it has a similar shape to the curve above, but it's just shifted towards the right because there's an incubation period or a period where there are no symptoms. So it takes a while from the HIV infection to the opportunistic disease.
That is so crazy.
Yeah.
It's so interesting.
Yeah.
Okay, let's see how much marks there are in this. 4 marks. So maybe.
Maybe give them a bit more.
Yeah, give me some more marks.
You could you not like just compare the high points of this graph and the high points of the other graph and say that's
you
probably
could like compare stuff.
around.
Yeah.
They always give you marks, right, for
For
stating
comparing stuff.
data,
Yeah,
like
they-
data points.
Especially when they say in the question, use the information given to explain the relationship. So they're asking you to tell you when does it peak in the first graph it's 1997, when did the deaths peak, it's 2004 or whatever that is. And then you explain it. And you can always also link that into
Could you also say that there was, like before 1990, there wasn't a lot of people infected with HIV because the death rate is.
okay, so
Is that a point? No.
well, you tell me.
Because it says use the information.
No, because we might not have known that people who died in 1990 at the time did have HIV because they couldn't you couldn't. So maybe more people did have HIV, but we didn't know that they died due to HIV because we didn't have like the means to sort of diagnose them
Really?
and like know
So-
their deaths. I don't know.
So then it wouldn't be on the graph.
I'm just saying that there could be a lot more people who had HIV, but we didn't know that. And at the time when people were dying, we didn't know that they had HIV, and we couldn't figure out that their deaths were caused by HIV. So that might not be a.
That's a fair point. If I told you guys that HIV has only existed since the late 70s, early 80s, does that answer your question?
Yes.
I remember watching something actually relevant. Okay, do we want to go to the mark scheme, or did you want to?
Your choice. Do you guys want to take a look at the actual marking schemes?
Sure.
Let's see what they say. Yeah, good.
There's a lot of points there for 3.
Mm-hmm.
So you get at first when somebody's first infected by HIV, they start showing signs of fever, and that's pretty much the only symptom
So yes, it's
for.
So it's basically an inflammation response in the blood. So when you first get the disease,
Because you're getting you have less T cells. No.
so the very first thing that happens is HIV comes into your blood and it goes into your CD4 positive T helper cells. When it's in those cells, the cells recognize that they're infected and they signal killer cells to come and kill them. That inflammation, that acute response, is what causes the illness, the fever, the malaise. That's very short-lived.
Okay. That's why there's fluorescence.
And-
so it's not.
Yeah, and so at that point in time, you have an increase in killer T cells and you have an increase in antibody against HIV because
It's an immune response.
you're getting an immune response. All immune responses will cause fever and that kind of thing.
But why does the immune system stop?
So it will go into a memory phase where it's constantly producing those antibodies and the B cells are circulating. But HIV is very sneaky because it can essentially hide in cells. And so the end of the acute response is the point which HIV has been removed generally, but it's become latent in CD4 T cells, where it's in the T cell,
But
it's
it
not
has an
replicating. It's not replicating, but it's in there, and because it's not replicating, the cell can't detect it, and it can't.
Okay.
So it can't
It's just
signal
waiting.
for itself
Why
to be
does
killed.
it just waits? So all the HIV guys go in. First, they start replicating in some of the T cells, which causes the immune response.
Mhm.
But then they wait it out for a couple of years.
So the HIV itself is constantly replicating. The thing that changes is the human's ability to control the infection. So if I draw a graph for you guys, I'm going to draw a graph, and on it there's going to be 2 lines. One is going to be your CD4 cells in HIV, and the other is going to be the HIV particles that go in. So what's happening here is at this point you're infected with HIV. HIV immediately starts replicating. Here is where you're going to get your acute illness. Okay, your CD4 cells are being massacred by your CD8 killer T cells.
Do I need to know that the CD4?
Is CD4 and CD8?
Just T killer cells and.
You don't need to know like a lot
Kills
about this.
Yeah, okay. Sorry.
But it's cool.
No, I'm just making sure because like I haven't done too many past papers in the mark scheme, so I don't know what exactly I need to know.
No, fair enough. Sorry, guys.
No, it's fine.
If this
So
is
this is like it's interesting, but
okay, I'll just finish this and then we'll move on.
Sorry.
No, it's fine. So in this period, you're let's not use CD8 and CD4 instead. Just use killer and helper.
Yeah, all we know is that it like infects T helper cells.
So your killer cells are killing your helper cells, and then your helper cells. So your helper cell population is going down while they're being killed, and at this point, HIV becomes latent. So there's enough HIV in the blood that it can be widely distributed, and it's not all replicating at the same time.
Okay, some of it's just not replicating.
Yeah, and that's normally what HIV does.
Why does it start replicating?
So it is constantly replicating at a low level,
Okay.
but during this period, the thing that keeps it down is these killer cells and antibodies.
Being up.
Yeah.
So how come
Our killer cells and antibodies decrease here.
So as the infection progresses, what HIV is doing is making these destroy helper cells. So at this point here, so many helper cells have been killed that they can't signal the killer cells to kill them anymore. Does that make sense?
Yeah. Okay.
So
So the helper cells can't signal to kill cells because they're all dying out.
they're dead, yeah, because the killer cells
So there are no more helper cells.
Yeah, exactly. So for the 10 years, killer cells and antibodies.
So they don't need kill cells anymore. Like at this point, no, you don't need more killer cells, so the body doesn't produce as many.
No, at that point, the body can't tell killer cells that it's infected anymore.
Because it's the
Oh,
T helper
okay.
cells that produce cytokines, which activate the T killer cells.
Yeah.
You
Oh,
need to
okay.
know that.
Okay. Yeah. I know that. Okay, I get it. So there are no cytokines released, so no killer cells activated because all the helper cells are dead.
So they can't. kill
Got
the
it.
infected
Got
cells.
it. Okay.
And
cool.
then, so at this stage, we have very low helper cells, very high HIV virions. So what kills the individual?
An infection that comes along, and there are no cells in the immune system an immune response can't be generated.
Exactly. So
Immunosuppressed.
yeah. So that's the
And it's an
Opportunities
autoimmune
to-
disease, right? That's where the kill cells are killing the helper cells.
no. So AIDS
Oh.
is this here. Everything after this is AIDS. So up until this point, you're an HIV patient. After this, you're an AIDS patient.
Is one of them an autoimmune disease or not at all?
So
Oh,
what does
okay.
AIDS stand for?
I don't know. Oh, autoimmune disease. Okay.
Autoimmune deficiency.
Deficiency.
Yeah.
Okay, I get it. I think. Cool, but that's good. Should we go?
Yeah, let's
Okay,
move on.
cool. No, it's my fault.
No.
Okay. A nucleotide is this, and this together. So those three.
Where's the phosphate?
Phosphate's this one.
Okay, inner replication.
So DNA is unwound and unzipped. Hydrogen bonds break between the strands of the DNA. DNA polymerase forms the sugar phosphate backbone of the two new strands formed, using both strands as templates. The free nucleotides bond with complementary bases with hydrogen bonding. You end up with two DNA molecules made from one DNA molecule, and both the DNA molecules consist of an old strand and a new strand, which is why it's called semi-conservative replication.
see where they allocated the marks.
What is is helicase the one that unwinds?
Yeah.
DNA helicase?
Unwinds it.
Or just DNA helicase? I don't know how to say any of them. Semi-conservative replication.
So, like this, so there are 3 nucleotides in the nucleus itself, right? And then the enzyme goes along the strands, right?
Mm-hmm.
And it catalyzes the reaction to form like phosphodiester bonds.
Yeah.
But I guess like my question is, how does it know when to add which nucleotide? Because it has an active site, right?
Mhm.
Which is always the same.
Yeah.
But nucleotides, which have to be specific to complementary, which have to differ in some sense. But
Is it maybe, I don't know the answer, a trial and error sort of thing where which one never matches because one has 2 hydrogen bonds and one has 3 hydrogen bonds? And only one that fits.
Yeah, so what determines complementarity? What determines what base?
well, it has to be a purine plus a pyrimidine.
And you need to have 2 hydrogen bonds and 3
Yeah.
hydrogen bonds.
Yeah, good. So that it's a physical restriction that creates the complementarity. So if DNA polymerase is sitting on a nucleotide and it wants to add another nucleotide, it can try adding each one. What's happening in real life is this reaction goes at a rate of like 1 million base pairs per second. So it's stochastic events where so many nucleotides are colliding with the enzyme and the right one is going to be put in place because it's the only one that can go in place.
So
That's a lot.
Yeah.
So now I think the tRNA has the codon, or is it the anticodon?
It's the anticodon.
Okay, the anticodon, and that consists of 3 base pairs, and so you have so this is happening in a ribosome, and you have an mRNA which has base pairs on it, and the tRNAs come along which have an amino acid on the one end, and then this anticodon which have 3 base pairs, and they bind with its complementary base pairs and start to form a polypeptide chain. So then the second tRNA's amino acid, which has the next 3 complementary base pairs, will a condensation reaction will happen between those 2 amino acids, and the polypeptide chain will start to form.
Yeah, good. Just be careful with your nomenclature when you're using the term base pairs because free mRNA and free tRNA aren't base pairs. They're just 3 nucleotides. And then when they bind, they become base pairs.
Okay.
That makes sense.
Okay, so the base pair is the 10.
Yeah, yeah.
Okay, cool.
But otherwise.
And I use nucleotides, so it has 3 nucleotides. Cool. So at the end of a tRNA doing its job, do we form another strand of something? Because you get the polypeptide chain, but you also get a double-stranded something, right? Do you?
So you mean the codon-anticodon interaction?
Or do they just like move on without sticking?
Yeah, they just move on. So the whole point of that codon-anticodon is to be, as the name implies, a code for
Awesome.
which polypeptide, which amino acid to break.
So it's not.
Yeah.
We don't have to do
We don't
this.
need, yeah,
Good.
So let's not break this. I'll just mute this. Okay. 2016 now. So Grace, talk me through this.
2016. Oh, sorry.
What? What? Which paper we're doing? 970 is
I wanted to know if it was.
Verse 3. November.
I was gonna ask if this was, but it's not because it's not 2017. Okay, fine. Just laugh at me. Okay, I'm gonna stop. Double absent in animal cells that produced what's it called? Chloroplast.
Good. Next one.
I'm guessing it's not cell membrane. I don't know.
Chloroplast.
Yeah.
So this is the one around the vacuole itself.
But it consists of a phospholipid bilayer.
Cool. All membranes.
Yeah.
Spindle.
Good.
A bacteria cell wall.
Yeah. Good.
Cytoplasm of an animal cell.
It's not actually the cytoplasm. Ribosomes.
Is it on the rough endoplasmic reticulum?
No, don't worry about it. This is one of those that you either know or you don't. It's the nucleolus. So nucleolus is, darkly shaded area of the nucleus that you often see in EMs, and its main function is to synthesize and assemble ribosomes.
So we don't have ribosomes in our cytoplasm?
We do, but were
That's
they
what
assembled?
Oh, okay, sorry.
Yeah.
So they're made in the nucleolus?
Yeah.
Okay, cool.
Okay, more.
Okay. So the viscum tubing would just be like swollen.
Good.
Can I ask something? So sucrose can't leave this, right? The viscin tube. It's too big to get through the viscin tube. Only water can only get in. Okay.
So yeah, you can literally just explain.
Well, there would be a mass change.
Okay.
It would be greater because you've got water coming into the viscin tube. That's happening because water's going down. So there's a high concentration of sucrose inside the tube and a low water potential inside the tube. So the water's going from a high water potential to a low water potential, which is why it's increasing in the sucrose until the potential gradient is evened out. There would be no other change. So no sucrose is going to leave.
Yeah, good. So sucrose is too large to leave.
Okay. Is that a point?
Yeah.
Okay.
But whenever you describe a process that has a name, write the name and underline it in the exam. Because
Should you underline like stuff like that? It just makes it easier for the examiners, doesn't it?
yeah, 100%. If you're certain, don't underline anything that you're trying to. Like, if you're unsure and you're sort of scribbling some words in, don't underline.
I've got a question. So, like, I'm quite used to not writing in bullet points now because I've kind of done it.
I only
for
write
pages.
bullet points.
Yeah, bullet
that-
points is good, but
Oh, okay.
should I, like, is it, do you need to do that?
You don't need to.
Okay.
It's not
But the only
necessary.
thing, like, why you do it is it looks better.
Uh,
Or it's easier for you to do, but it's not for the exam.
It also makes your answer more succinct and easier to read. So it
Okay.
is easier for the examiner
Okay.
because they would much rather just skim read 3 bullet points than, actually.
So they might be just like
Dissect your
really
grammar.
crossed and mark you a bit.
Hey,
well.
my handwriting is bad. They're not gonna make bullet points. They're just
This
gonna be-
student is just 5 marks off for just making it.
No, they would.
His sister then.
It's so great.
You'll be okay. You can make bullet points. Okay.
So what name should I be writing here?
Well, you tell me. What's the process for describing and studying?
Osmosis.
Good. Yeah.
Oh, okay.
There's like 4 different points you can make here, and each of those points are a component of osmosis. But if at the start you just write it's the mass increases due to osmosis, and then you just explain what it is. Okay. Mark.
Okay. When sucrose is actively pumped, when sucrose diffuses from the companion cell to the plasmodesmata into the sieve tube element, the water potential of the phloem decreases. So water enters the phloem sieve tube at the source by osmosis down the water potential gradient, increasing the hydrostatic pressure. Then at the sink, when sucrose leaves the phloem, water flows with the sucrose out of the phloem sieve tube, because the water potential has increased by the sucrose leaving. So the water potential flows through osmosis down the water potential gradient.
Good.
Thus, the hydrostatic pressure decreases and it maintains that difference for mass flow to occur.
Good.
Can I ask something about the start? So at the start, when sucrose comes in from neighboring cells into the companion cell first, right? And that's actively transported or diffused? Actively transported. Sugar is always actively.
It's not the sugar itself, it's the hydrogen ions that are pumped in order for the sugar to go with, because once the hydrogen ions are pumped into the cell wall of the companion cell, they set up a gradient for the hydrogen ions to diffuse by.
Okay.
And the sugar molecules go with the.
So there's a proton pump
Yeah.
pumping it from the neighboring cells into the companion cell wall.
The companion cells are the neighboring cells.
So the companion cell cytoplasm to the companion cell wall.
Okay.
Yeah.
And then because of that, facilitated diffusion occurs with sugars and what?
Water follows.
Through a
What goes-
co-transporter protein.
Okay, so because of the protons being pumped into from the companion cell cytoplasm into the companion cell wall,
Mhm.
facilitated diffusion occurs with the assimilates the sugar, and then the water potential, and then that moves by facilitated diffusion into the phloem, or
Diffusion, because there's plasmodesmata, so cytoplasmic connections binding the companion cell of the sieve tube element together.
Told you.
Yeah.
Okay. So you hit all the marks that they were looking for with that explanation. Let's see.
Do you want me to draw it or?
Yeah, go ahead.
I asked for this in a paper that I did today.
Aren't you lucky?
Mm.
No, wait. I need to think fast. Dun. No, I'm right. Okay. So I have this. And then just like the rest of the molecule here and here. And that's so this is the peptide bond.
Mm-hmm.
Sorry. Do I need to do anything else? Oh, do I need to say that this is a condensation reaction so that you have water as well?
Yeah.
How many marks is this?
2 marks.
Oh, this text space. Okay, so I could say that this is a condensation reaction and that this is the peptide bond.
Yeah.
So no. Glycosidic bond and peptide bond. Glycosidic bond is which one? Is that in biology or is that only in chemistry?
No, glycosidic bond is like between sugars. Well, yeah, like in the polysaccharide, you have a lot.
Okay, and the peptide bonds are between amino acids.
It's like that oxygen.
Which is the same as an amide. Okay. And the glycosidic bond also releases, it's also a condensation reaction.
Yeah, because like that.
But you have
proteins
COC.
It's like all of them, like the ester bond between glycerol and fatty acids, condensation, peptide bond, condensation, glycosidic bond, condensation reaction.
Okay.
You can think of it as anything that's building up material is condensation, anything that's breaking down is electrolysis.
Okay. And with the glycosidic bond, so for example, between the fatty acid and between glycerol, it's COC.
Oh, you're saying that it loses OH.
So the glycerol has an OH on it, and this has a carboxylic acid group. And the OH from the glycerol and one of the H's go away, and it bonds together with the oxygen. Okay, cool. You know it.
Huh?
That's okay. Cool. Fructose is a pentose sugar, so it has 5. No. Sorry, they both have the same formula, but it has sucrose they have a different shape. One is of an octahedral, I guess, the 6-sided shape, and the other one's a pentose shape. I don't know.
Not quite. So when we classify carbohydrates, what are the different kinds of carbohydrates?
Oh, sorry, wrong. Okay, I'm going to see. Fructose is a monosaccharide, and sucrose is a disaccharide, and sucrose is fructose plus glucose. There you go.
There you go.
Do I need to say anything else?
So in terms of structural differences,
Like
So-
anything that can be different.
Huh?
Anything that's different.
So in fructose there's only one ring, and in sucrose there are 2. And in sucrose has more carbons.
it does.
Does that count as a structural difference?
Yeah, I mean they'll give you the mark if you write the formulae.
Okay.
But I mean you don't need to go into that kind of detail necessarily. As Mark says, you pick up the marks for just saying the difference in number of rings, the fact that one of them has a glycosidic bond and the other doesn't.
Okay.
Okay. And out of curiosity, if you wanted to write out the formulae, how would you do it?
So for fructose, I could just write C6H12O6, and for sucrose, it would be C12H22 O30. No, sorry, O31. No, sorry. O11.
Yeah, exactly.
Yeah.
So just times by 2 minus H2O. Okay, good. Okay. So
Oh, okay. So the enzyme that has an optimum temperature of 95 Celsius can catalyze reactions at higher temperatures. So at higher temperatures, there are more frequent collisions between the enzyme and the substrate. And this because it has a higher optimum temperature than 37 degrees. So the other enzyme would denature. So that means that the bonds would the atoms would vibrate so much that the bonds break and the active site changes and the substrate can't bind to it.
Yeah, good. So key terms here, denature. You could talk about Vmax if you wanted to say that.
Okay.
Thermophilus would be able to achieve a higher Vmax because it can operate at higher temperatures, and that way you have. And then you talk about the advantages in terms of yield, so you can produce more high fructose corn syrup faster.
Yeah.
It's very chemistry.
Or like more enzyme substrate complexes form.
Yeah.
What are the marking points?
So this one is idea that you can use the higher temperature. Increased temperature increases collisions or number of ES complexes. More product / higher rate of reaction. Less prone to denaturation. More stable / less stable.
Cool.
Okay.
Can I go down? Okay. So I could say that the immobilized glucose isomerase and the immobilized enzyme has a higher optimum pH than the glucose, the free enzyme. Oh. Yeah. I could say that they both increase up to that point and then start to decrease as the enzyme is. Does the enzyme denature when its pH is not optimum? I don't know. I could say that there's a higher, so I could compare percentage activities. So until when, for example, up till when the free enzyme has a higher percentage activity than the other one. Yeah.
Okay, so go on.
Is there a point for saying that they're both active from 6 to 9? Because that's the most basic thing.
Let's see.
Might be too basic.
I don't think
It's too basic.
Yeah.
But I mean, I would still
that down.
just for fun.
because often,
You never know.
yeah, you really do never know, and often the first marks are always going to be the simplest to get. So when the examiner compares what you've written to the marking scheme, so they often mark 100, 200 papers in one session, and they'll have marking scheme. They'll mark question after question after question, and so they know the marking scheme in their head. And if they look at your paper and you've just done it in the simplest way possible, they're just going to be taking it. Yeah, it's what they want us to do.
Not with my paper.
Because they're gonna have to read everything.
I have to read it.
Yeah.
All the useless ands.
Yeah, literally.
One of my friends actually had to rewrite an exam essay.
Really?
Because they couldn't read it.
They couldn't read it.
But that's so nice that they actually let him rewrite it.
Yeah, I mean, they weren't happy about it. There you go.
But I'd be scared they'd just be like wrong because I can't read whatever they've written. And I'm
Yeah.
sure they actually do that for some Actually, I think because it's not
I
like
hope they don't do it for me.
no, but your handwriting is still legible.
Yeah, I wouldn't worry. My handwriting is terrible. It's never happened to me. But anyways, yeah. So what they talk about in this is peak activity. So always use the actual values. You know, peak for this is there, peak for that is there. Then talk about how from 6 to 8 pH, free in solution enzyme has higher activity than immobilized enzyme, and from 8 to 9, immobilized has higher activity than free. And you can talk about the increase and the decrease. You can talk about the steepness of the inclines.
So, like, you would probably get a point for talking about the steepness, but how would you say is the easiest way to show that with data?
You could calculate a gradient, but you could just say that this one's steeper than this one.
Just observe it. Don't calculate the gradient.
Unless you've got so much of free time.
You will, but don't bother. It's like they don't even want to see that.
Yeah.
If you want to make him a bit more crass, let him do the maths to see if he got this.
All right. So, Mark.
There should be
There's
another one, right?
Yeah.
Here, okay, so how do you describe this?
Okay. Right. The first one's correct. The second one's correct.
Mm-hmm.
Do you have to go through this?
Do you follow?
I am confused. Like I know actually what you. Give me a second.
Take your time.
Oh, okay. Okay, so it's AUG, like that.
Yeah.
Okay. Wait, student asked to work out. That would be. So correspond, they're not asking for the complementary, they're asking for. So we don't need to first make this T, sorry.
You tell me.
Look at the mRNA nucleus. That would correspond to the first 5 amino acids.
What are you talking
I feel
about?
like it would be wrong.
Okay, so I know where you're going from, and this is actually really important because these can be the more trickier questions.
I'm
thinking it's the anticodon.
Yeah.
Yeah, and you need to be sure about it, or you're gonna get, it's just 1 mark, so it doesn't matter really. But in other questions, it can be like quite a bad effect if you make this interpretation wrong. So what my advice is read the question very carefully, because the question is going to say codon and anticodon. The question is going to say mRNA and tRNA, and if you know exactly what the data they're giving you is, and I would literally write, so they say student was asked to work out mRNA nucleotide sequence, so we know that this is mRNA. Write it out so that
And it says here mRNA codon, so it's
the
the same.
table is mRNA. Okay. Exactly, yeah, so it's the same.
If it was anticodon,
Mhm.
so would they say tRNA anticodon?
Yeah,
Or
they
would
could say,
okay.
they could say.
So if one was the other, then I'd need to change it.
Yeah.
Okay, cool.
And sometimes they ask you to compare DNA to articles.
And then I need to be careful with you.
Exactly. It's the exact same apart from UT.
Cool.
But yeah. You happy with that?
Yeah.
Okay, cool. Next one.
Could it be because we have the same combination of base pairs sometime, different combination of base pairs sometimes equal the same amino acid?
Exactly.
And yeah, so do bacterial cells only have specific amino? No, sorry. So yeah, there are different combinations of base pairs that will code for the same amino acid. So the bacterial cell might have a different codon, which still will give you the same. I don't know what else to say.
Okay, so the enzyme under study here, glucose isomerase, you can assume that it's not. You can assume that this is like going to be a human enzyme, right? And they're asking you how the nucleotide sequence worked out corresponding, blah, 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 may not be the same as the mRNA nucleotide sequence for those amino acids present in the bacterial cell. So one thing is redundancy, which you've explained.
Redundancy.
Yeah, so sorry, that's the word given to the mechanism you just explained. So the fact that multiple codons code for the same amino acid is redundancy. Why might we have redundancy?
Could it, so that we can have, maybe to count for if errors occur?
Good, so it protects against devastating mutations.
Does it have something to do with maybe the bacteria has different tRNA? So like only specific combinations of tRNA, and they might not have the same.
Yeah, that's a good point. Looking at the marking scheme, the marks that you get, so you get one for talking about the genetic code being degenerate or redundant, and the rest of the marks come from explaining what that means. So, 64, you can have 64 different types of codons and you only have 20 different amino acids. The idea that more than one codon specifies an amino acid, example of choice of codon to specify the same amino acid. So they've literally just taken one mark for, describing what it is, and then more marks for sort of explaining it, but I think it's just describing it.
Okay, so with questions like that where there seems like one obvious answer, I should explain them.
Yeah. Especially, he says, this says discussed with reasons.
Okay.
And it also specifies with the first five, so yeah. That question I don't think is very well worded, to be honest, but I think he would have picked up the marks anyways, so.
This is an artery because it has a thick tunica media. The tunica intima is not smooth, it's wavy, so it's also an artery. Yeah. They said one actually.
Say
so
that
the tunica.
the lumen is not that big?
You could say that.
Could I say that from looking at that?
Yeah, you could say that. Technically, you can't say that because you don't have a scale bar, but
Yeah.
you would get a mark for saying
Okay.
that. Because that is a feature of arteries.
Cool. What happens, okay, so when they say 1 and you say 3.
Don't do that.
Don't do that?
Yeah,
Really?
definitely don't do that. If it says 1,
Say 1?
Only one,
Okay,
yeah.
because my other biology tutor said, just like,
No.
Yeah. No, because in the mark scheme, especially like in A2, sometimes they'll ask you for
like there was there sometimes they ask you for questions, but if you don't say specifically what's in the mark scheme, then you might not. It's just
But
it's
like,
confusing.
what's the point if they said like 1?
I think
Do
they've.
like 2.
I think they've. I thought they underlined it, like made it obvious, because so that people know that they don't have to do more than 1 or more than 2, sort of thing. Not because.
It's because like if you do so many and if you're not sure, then 1 is bound to be.
Oh, yeah.
It's not-
Well, let's see. Okay, I won't do that. And what if all of them are right?
I mean,
Well,
they
actually-
would give you the mark if you put down those reasons and they're all right, but you're just increasing the chances of
Of
yourself
saying
being wrong.
Yeah, but if you okay, it's
There's
good.
no-
It's also,
Excellent.
it's also a time sink as well.
Okay.
You want to like be getting through these as fast as possible. If you're stuck for more than a minute on a question, move on and come back.
That's why you don't want to write bullet points.
But these papers aren't that bad.
I know, I
mean,
finished them.
explain the relationship structure for tunica media. Okay, so renal artery. I don't need to explain that, but
Okay, wait. So tunica media, the reason why it's big, is because of the high blood pressure inside of the artery. So it won't, for example, burst. So it can I can't remember the word, but it can contain. It's able to withstand that amount of blood pressure. Also, so that the blood pressure is the blood is so that so you need that high blood pressure so that the blood can circulate throughout the body. Can I also say that it's okay for the renal artery, because that's to the kidneys, since that's quite far away from the heart. It needs to, like what I just said before, actually. So it needs to maintain that to be able. Does it also help? Does it also help push the blood along so that it can actually get where it wants to get?
Okay, so the only thing that pushes blood is the heart.
Okay.
Don't mix yourself up there. You're fine. I'm sure you'll get the marks for that. I'd be careful here with answering the question specifically. So they're saying structure of the tunica media. What gives an artery its ability to withstand pressure?
It's elasticity.
Yeah, and where does the elasticity come from?
From its tunica externa.
Yeah, so loads of elastic fibers. And in big arteries like the aorta, the tunica media will be packed with elastic fibers as well. So that's fine.
Okay.
You're on the right tracks. But if we look at this vessel, which is an arteriole, so this is a smaller artery, and you can tell because of the ratio. You don't need to worry about this, but I just want to explain it to you guys. So there's so much smooth muscle here, and there's very little tunica externa. So this is true for the general structure of the tunica media and the general functions of the tunica media. Lots of smooth muscle layers means that it contracts and relaxes. And
It
that's
controls the volume of the blood flow.
good, yeah. So that's the function. Muscle, whenever you see muscle, it only does two things. It either contracts or relaxes.
Okay.
And if you see loads of muscle in a blood vessel, that blood vessel must be regulating blood flow. So by contracting, it reduces blood flow to an area. By relaxing, it increases blood flow to an area.
It's the
Okay.
same with bronchioles.
So it's nothing to do with pressure.
No, I think you still get the nostril pressure.
Okay.
It has a thick wall.
Let's see what they say. Yeah.
But I should say, when I'm talking about blood pressure, I should talk about the elasticity, which is in mainly the tunica externa.
You talk about like it having a thick wall, able to withstand pressure, but you're also talking about it prevents the elastic layer prevents it from bursting,
Mhm.
so it allows the artery to like
Expand.
give to the pressure
Yeah.
by expanding. And you could maybe say that it smoothens the blood flow.
Why is this?
elastic tissue.
I didn't know that the tunica antenna was wavy. Why is it wavy?
I
So
don't know why that is.
How does it make that? I thought the tunica antenna was to make sure
Arteries
that
are wavy.
resistance is
Sorry. Brings us like
I thought as least possible friction.
at the end of yeah.
So how would something being wavy be?
it's not functional. What you have is when your arteriole is maximally dilated, you have
okay.
a monolayer that's smooth, and when it contracts, it becomes smaller.
So
The
less.
cells themselves are going to become wavy because they're packed in that way.
I get it.
The cells can't shrink. Once
Is it the same for the lymph? Because the lymph also has smooth muscle that contracts a bit. But
Yeah, lymph is closer to veins in terms of structure.
That is so crazy. I didn't think of that. Okay, so and it needs it to be smooth when it's expanded because that's when it wants the blood to rush through. And I guess when it gets smaller, you probably also have higher resistance because of the waves, which means less. That's also probably bad.
Yeah,
Okay, cool.
decrease blood flow.
Awesome.
Very good.
What does it say in the landscape side?
So, does it say? Okay, come look. So you can't say that muscle stretches. Or
You can't.
stretching and recoiling because that's a property of elastin. So they
What?
elastin, so the elastic tissue. So
Which is only in the outside
no, it's also in the tunica media, but it's mostly in the
Okay.
external. So I mean, the key for this is don't mix up muscle and what it does and elastic fibers and what they do.
Okay.
So you know what happens. You're getting the marks right. Just don't mix up muscle and elastic fibers. So they say here, muscle increases blood pressure or maintains blood pressure. Elastic tissue
And
withstands
withstands.
high pressure. Elastic tissue smooths out pulsed
But-
flow.
Oh, okay, because there's elastic tissue. Okay, I get it now. Sorry. And then that's the altering volume that you were talking about.
Yeah.
Cool.
Cool. Shall we stop there, guys?
Yeah.
All right.